Our next speaker, Dr. Melody Zeng, did her PhD at Indiana University and her postdoc at the University of Michigan, where she worked closely with Gabriel Nunez. She started her own lab in 2019 at the Weill Cornell Medical College in New York City, where she is an associate professor of immunology in the Department of Pediatrics. Her lab employs multidisciplinary approaches to identify and define gut microbiota-regulated immune pathways to advance therapeutic development for diseases that stem from gut immune dysregulation in early life, including neurodevelopment, cancer, infection, and allergies. And so with that, please take it away, Dr. Zeng.
Thank you so much, Alex, for the introduction. Hi, everyone from New York City. I figured there'll be a lot of people from different parts of the world. Thanks for tuning in. My lab's been doing a lot of work addressing the crosstalk between the gut microbiota and the immune system, as Jun's beautiful talk on early life microbiota and beta cell development highlights. The microbiota, especially in early life, is really critical in shaping the immune response in early life. And that's a major reason why when I started my lab, I wanted to study this context, because this may be a very important critical developmental window where we can identify microbial targets that can be harnessed to improve immune system development. And let me see. There have been a lot of studies probably over the past decades really underscore the importance that the development of the immune system occurs very early on in life, as early as in utero development, where the conditions from the mom would be very critical in driving the immune system in the fetus. So during this time, it could be the mom's microbiome, it could be the mom's diet, and also the immune status that can influence the microbiome in the mom, and the metabolites from the mom can cross the placenta and reach the immune cells in the fetus and shaping the fetal immune cell development. And now after birth, there'll be unique bacteria that are colonizing the newborn's intestine, which now can continue to shape the immune system development in the newborn. And Jun just showed the microbiome in the newborn affecting beta cell development through regulation of macrophages. There have been also studies showing that the metabolites in the newborn can have a huge influence on skin sensitization to allergens, and especially in the context of atopic dermatitis. And in the lung, changes in the metabolites from the newborns have been associated with increased risk of allergy in kids. And now there's been a lot of interest looking at the gut-brain crosstalk, and particularly in understanding how that might be a very important underlying driver of neurodevelopmental defects such as autism. So Jun also showed really nice background on the changes of the microbiome in early life and how that might become stable around 2 or 3 years of life. So now when we look at the newborns' gut microenvironment, we would see a little higher oxygen, and the nutrients are mostly derived from maternal milk, including lots of simple sugars and human milk oligosaccharides. And at the same time, there will be very little complex fibers because they're not eating solid foods yet. So the combination of the different factors here in the microenvironment would really determine the types of bacteria that are able to live and thrive in that environment. So in the newborn's intestine, you will see a lot of lactobacillus and enterobacteriaceae, which are able to proliferate in the presence of little oxygen, and they thrive on simple sugars. And later on, as the oxygen level goes down and increasing levels of complex fibers, there'll be other bacteria that are going up, including bifidobacteria, Clostridia, and Bacteroides also. And later on now, by the age of 2 or 3, we will see pretty stable microbiome in the toddlers. That's similar to that in adults. So just to highlight the importance of microbiome, especially in infant, I'll just start off showing a little bit of our project looking at human infant. And so my lab has been collecting stool samples from babies from the neonatal ICU here at New York Presbyterian in New York City. And we've been very fortunate to get a lot of support from neonatologists here. So with the samples, we typically study the composition of microbiome and study functional changes of the immune system by transplantation of the bacteria into germ-free mice. And just to show you a little bit of our data from a project that is still in development. Hopefully we'll have more to share on this project. So here we wanted to see how the perturbation of the microbiome in preterm infant might be contributing to what we usually know about preterm infants when they're dealing with respiratory viral infections, because preterm babies are born with smaller lungs and a lot of the organs are underdeveloped at the time when they're born. But is it because of immaturity of the development of the lung, or is there any contribution coming from the altered microbiome in preterm babies? So here, if you look at a cohort of preterm babies versus term babies, so here the preterm babies were just about 4 weeks premature at the time of birth, versus the term babies, we do see significant changes of the microbiome in the stool samples, where in the preterm babies, we will see a lot of Veronella, Enterococcus, as opposed to increased abundance of Bacteroides in the term babies microbiome. And that's pretty consistent with some other studies. And now to see whether the altered microbiome in preterm babies might have some role in changing the immune response to viral infections. And so we transplanted the stool samples from a consortium of preterm baby microbiome into germ-free mice. And we did the same with another cohort of mice with the term babies microbiome. And now the gnotobiotic mice would be infected intranasally with RSV. And now 4 weeks after that, if you just look at the lung, you will see immune cells infiltrating into the lungs in both groups of mice. But importantly, we see significant increases in the immune cells in the lungs from the mice with the preterm microbiome. And the mice here were all the same. It was really the microbiome that we transplanted into the mice that were different. So really highlighting the microbiome in the preterm babies might be linked to the dysregulation of the immune cells to RSV. And now when we look at the immune cells in the lungs of those infected mice, we will see a reduction in interferon gamma T cells, which are critical in dealing with viral infection. And specifically, when we looked at the RSV-specific CD4 cells, we will see a huge drop in those cells in the lungs from the mice with the preterm microbiome following RSV infection. And that's consistent with the reduction in neutralizing antibodies in those mice compared to the mice with the term infant microbiome. And to really appreciate, so this really suggests that there might be impairments in developing protective, adaptive immunity following viral infection in the mice with the preterm microbiome. And that might be consistent with what we see in preterm babies. Usually when they get infection, if they are not really generating protective memory T cells or B cells, they might have trouble dealing with the subsequent infections. And now to test that, we did a reinfection in the mice to appreciate the recall response. And now you can see in the lungs from the mice with the turned infant, you see very, very little immune infiltration, suggesting here now we're actually using a really high dose of RSV, suggesting protection by the immune memory cells from the prior infection. However, now, if we look at the lungs from the mice with the preterm infant, we see massive infiltration of immune cells in the lungs, really suggesting they were really not getting help from the immune memory that should have been developed from the prior infection. So we're still doing a lot of mechanistic studies to really appreciate the factors coming from the microbiome in the preterm babies that might be skewing the immune response to RSV. Hopefully we'll have more to share in the near future on the project here. And the first talk of the day, Brittany's talk, she also highlighted the connection between the gut and the brain and neurodevelopment. And we also stumbled upon some gut metabolites that support that. And here we profile the metabolites in the young mice early in life. So here we're really looking at P14 mice, specifically in the small intestine. So the paper's been published, so I'm just showing a little bit of the data from the project. And so here we profile about 500 metabolites, compare the metabolites in the adult and neonate. As shown here, you can see a lot of the metabolites that were either elevated or reduced in the neonatal mice compared to the adult mice in the small intestine. And this work was done by my postdoc, Catherine Sanderdad, who's been really productive in my lab. And specifically, if we look more into the metabolites that were elevated in the neonatal small intestine compared to the adult, to our surprise, we saw the metabolites that were severely elevated in the neonatal small intestine were neurotransmitters, including serotonin. And neurotransmitters are usually produced by endochromaffin cells at least in adults, in the gut, and the gut is known as the second brain in humans because the gut is responsible for making more than 95% of the neurotransmitters in our human body. But now we were really surprised to see a lot of neurotransmitters already produced at such a young stage of life, at least in mice. And we validated that with the studies of human infants as well. So now we did a bunch of studies and really excluded the host as the primary source of serotonin at least in day 14 mice. And we have additional data showing that it was really the unique bacteria that are enriched in the neonatal intestine that were the producers of serotonin, also known as 5-HT. So we isolated a huge number of bacteria and tried to measure serotonin in the splenotins and in the bacteria that we isolated from young mice and human infants. We were able to detect quite a large number of bacteria generating serotonin. In mice, we will see Rhodantobacter haley and also Enterococcus gallinae as the major producers of serotonin. And human infants, we found Staph aureus among the major producers of serotonin in human babies. In the interest of time, I'm not showing a lot of the other work from the paper, but what we discovered was that the serotonin in babies mainly comes from bacteria that are uniquely colonized in babies. And now we also have data indicating that serotonin directly signals to naive T cells where it gets into the cytosol of the T cells and converted to indole-3 acetaldehyde, I3A, which now inhibits the mTOR activation, changing T cell metabolism. And this leads to the differentiation of T cells to regulatory Tregs as opposed to pro-inflammatory Th1 or Th17 cells. So now the Tregs can be used to suppress T cell reaction to food allergens and commensal bacteria that are colonizing the infant's gut during that early stage of development. So the data really highlights that it's very important to have the right types of bacteria during that early developmental stage. And the serotonin produced by some of the bacteria that are enriched in babies can be co-opted by the host to shape the T cell response and promote immune tolerance in early on in life. So moving forward, we also are interested in looking at the microbiome in immune system development at an even earlier stage of life, and specifically looking at in utero development. So this is a project by my postdoc, Julia Brown. She wanted to see during pregnancy whether the microbiome coming from the mom might have any influence on the immune response at the maternal interface. So Julia first looked at germ-free mice during pregnancy and compared that to specific pathogen-free mice. And when we looked at the mice at E16.5, which is probably 2 or 3 days before birth, we were very surprised to see a lot of resolved fetuses in the germ-free mice, as shown here, compared to really nice looking embryos here in the specific pathogen-free pregnant mice. And as shown here, we will see a pretty significant resorption rate in the fetuses from germ-free pregnant mice. And at the same time, when we measure plasma anti-fetus IgG, we will see really high levels of plasma IgG in the germ-free pregnant mice against fetal antigens, really suggests loss of maternal fetal tolerance in the absence of gut microbiome. And that's recapitulated when we treated the mice with antibiotics, where we saw increased fetal resorption, similar, although not as severe as in germ-free mice. And now we wanted to further appreciate immune dysregulation, what might be driving the loss of immune tolerance in the germ-free pregnant mice by single-cell RNA sequencing of immune cells in the placenta and also uterus and blood. Here I'm only showing the placenta single-cell RNA-seq data. You will see multiple immune cell populations that are altered in the germ-free mice compared to the SPF pregnant mice. And here you will see a diminishment of myeloid-derived suppressor cells, changes in dendritic cells, really interesting data here. And by analyzing specifically the T cells, overall, we saw increased interferon gamma signature in the memory T cells from the germ-free placenta, and that's also validated when we did flow cytometry analysis of the immune cells in the germ-free mice, and you can see similar trends in the uterus as well, but not in the spleen, really suggesting the immune changes are more limited to the maternal-fetal interface. And to really appreciate the importance of the interferon gamma, whether that might be linked to fetal reinfection here in germ-free mice. So now we try to neutralize interferon gamma in the germ-free pregnant mice as shown here. You can see really nice rescue of the fetuses in the germ-free mice when the mice were treated with anti-interferon gamma systemically. And now try to understand what's driving the interferon gamma signature in the germ-free mice. We went back to our single-cell data, and one important thing that we noticed was the reduction in myeloid-derived suppressor cells in the germ-free placenta and uterus, as shown here, the MDSC cell population. And MDSCs are known as an immune cell population that would have more immunosuppressive effect. And even though developmentally they're similar to neutrophils. So here, when we use anti-LY6G to deplete MDSCs, also possibly neutrophils, keep that in mind, we saw rescue of the fetuses in the germ-free mice, and at the same time, let me try to see the anti-LY6G. We will see changes in the sorry, here this is the SPF mice. When we deplete the MDSCs, we will increase the fetal reduction in the SPF mice, and at the same time, we will see increased numbers of CD4 T cells making interferon gamma in the placenta and uterus, really demonstrating a possible link between MDSCs at the maternal interface and the interferon gamma T cells. And now to further appreciate whether it's really the reduction in MDSC numbers or functional changes in the germ-free mice, by looking at the gene transcriptome, we found that the germ-free mice, the MDSCs show, or neutrophils are showing really elevated signature of genes associated with antigen presentation. Even though the B cells or dendritic cells, we saw no changes in those genes compared to the SPF mice. We have additional data really showing, and now we when we isolated the MDSCs from the germ-free pregnant mice and transfer into SPF mice, we can see a huge increase in fetal reduction in the mice receiving the MDSC neutrophils, and that's consistent with increased numbers of interferon gamma T cells in those mice, really demonstrating MDSCs as the driver of the interferon gamma cells, which leads to fetal reduction. Consistently, when we looked at human placental tissues from women with recurrent miscarriage, we will see a reduction in MDSCs and at the same time expansion of neutrophils. And in those cells, we will see increased expression of genes associated with antigen presentation, really suggesting this regulation of MDSCs in human with recurrent miscarriage might be associated with the loss of maternal fetal tolerance. This is the last piece of data here to really appreciate metabolites coming from the gut microbiome that might be promoting maternal fetal interface. Here we profile the metabolites in the amniotic fluids and plasma in germ-free and non-germ-free pregnant mice. And specifically in the amniotic fluid, that's where we see a significant reduction in a lot of the metabolites associated with tryptophan metabolism, including kynurenine, indoles. And now when we supplemented the indoles here in the 3-carbonyl to oral gavage into the germ-free mice, we will see really nice reduction in the interferon gamma T cells at the maternal fetal interface, and then a reduction in anti-fetus IgG, rescue of fetuses, really nice rescue in the germ-free pregnant mice, and that's associated with the downregulation of MHC expression in the MDSCs in the germ-free mice. So really underscoring the connection between microbiome and then the MDSCs and interferon gamma T cell response at the maternal fetal interface. And lastly, this is really nice data when we profile the metabolites in human ductal tissues in women with recurrent miscarriage. Consistently, we saw a reduction in indels in the ductal tissues, really in some way validating what we found in the mouse system. And so with our paper reporting what I just shown here is going to be online next week, and it recently got accepted. And besides the MDSC interferon gamma connection, we also found some regulatory T cells originated from the intestine during pregnancy also migrate to the maternal fetal interface and contribute to that maternal fetal tolerance. And if you're interested, you can find that from our paper, which will be online next week. With that, due to time, I'll be happy to take any questions. Thank you so much for tuning in.
That was a great talk. It's cool to see just like so many ways that you can kind of approach the question using these sort of like multi-omic approaches. Yeah, I think we have some time for a few questions. Our first question from Sean actually asks or says, your mouse work indicates that the microbiota is required for proper fetal development. Is there epidemiological evidence in humans for adverse effects when you take antibiotics, when mothers are taking antibiotics during pregnancy? And maybe like, how does that impact the outcomes of pregnancy?
So I think pregnant women usually when they're treated with antibiotics, it's because there might have been some suspected infection. So I think that might be a factor that will complicate how we appreciate the importance of microbiome. And I think there have been studies showing that is actually an increase in at least pregnancy complications in women treated with antibiotics. But as I said, that could have been just because they already have some underlying conditions. And in our study, I did not have time to show, but we actually treated mice with vancomycin and gentamicin separately. And it was really when we treated mice with vancomycin where we will see a reduction in the indels in the amniotic fluid. And that's also consistent with increased fetal reduction in the vancomycin treated mice, and an increase in interferon gamma T cells and increase in anti-fetus IgG. So I think the mouse system is really nice because it allows you to address the microbiome alone without any confounding factors.
Yeah, that makes a lot of sense. I think, yeah, when you're doing these human population studies, it's hard to account for all the other covariates. Yeah, sort of a question that multiple participants asked was kind of along the lines of acknowledging that like the mode of delivery can maybe have a strong impact on the development of the fetal microbiome, and just kind of wondering if like looking at C-section delivery is something that you've considered, and maybe its impacts on the microbiome and the development of the immune system.
Yeah, I think there have been actually a lot of human studies showing changes of the microbiome in babies born through C-section or born vaginally. In general, in the babies born through C-section, you will see enrichment of bacteria that are more similar to the skin microbiome, possibly coming from the mom, versus the vaginal birth. The bacteria that first colonize the baby's gut would be mostly acquired through the vaginal, the birth canal from the birthing process, and there have been studies showing the babies born through C-section might be at higher risk of developing allergies and asthma later on in life, and in part due to the alterations in the microbiome during that early developmental time window. But there's been also some studies showing that, in particular in the babies that later on might get breast milk, that might actually help convert or reverse the microbiome, and by the age of 6 months, the microbiome in the babies through C-section might now look more similar to the babies born through vaginal delivery. So even though I think C-section is pretty common, a lot of people might have been born through C-section, but it doesn't mean it would permanently change the microbiome and permanently dysregulating in the immune system. I think there are some other interventions can be leveraged to kind of normalize the microbiome in those individuals.
That's fascinating. Thank you for sharing. And yeah, it definitely, I feel like I've seen other studies that really point to the importance of like breastfeeding and receiving like maternal antibodies through breast milk. Yeah, I think that's all the time we have for questions. Thank you so much again, Dr. Zeng, for joining us. And
Thank you.
I'd also like to thank Dr. Round for also joining this session. It was amazing to hear you guys both speak. And so I think that concludes this particular session. We're going to take a short 10-minute break, and then we will be back with the final session of the symposium. Thank you so much, everybody.